Okay, so welcome everyone today to our lunchtime session, and I'm going to be talking a little bit about sample requirements for cancer genomic testing, a little bit about what kinds of testing we do, and some of the sample requirements and why we ask for them in the way that we do and the impact this has on testing. Okay, so just a very quick background to the kind of things that we're looking at. We're looking for variation and mutations. A lot of what we are doing and analysing and trying to look for is small changes, so changes in a single base or changes of a few bases within the genome. And there are hundreds of thousands of these, as you all know, across the genome. And we're looking for ones that we then have to interpret whether they be significant and deleterious or change the function of a protein. We're also looking at large changes, so we can look for structural variations and large changes in the DNA and chromosomes. We can look at copy number changes, so we look for larger deletions, and we also look for gains and amplifications of genes. We look for translocations where material crosses between and within chromosomes, and you can get genes moving around and coming under the control of other genes. And we can also detect inversions where we get a piece of DNA almost taken out and then put back in the other orientation. All of these things can have effects on how the gene functions and how the protein is then working or not working. We use a variety of techniques. Many of them are PCR based. We have a variety of PCR based core tests. Basic PCR, we use a real-time PCR, which can be more sensitive and also allow us to quantify material. We use digital PCR, which can allow us to get down to very, very low limits of detection when we're trying to pick up really low-level mutations. We can visualize the chromosomes using fluorescence and FISH and changes and structural changes. DNA sequencing, I guess, is our mainstay. We do a huge amount from small regions where we can individually look at every base to do huge amounts of next generation sequencing where we can parallel sequence huge amounts of genes as well as many patients all at the same time, and then do some very advanced bioinformatics to actually look for the changes between the reference sequence, what we see as normal, and any changes in what we're sequencing. We can do tissue microarrays to again look for those larger structural variations, and then I suppose it's next generation sequencing, but of course we're now doing whole genome sequencing as well. So in our lab, as I said, we have many testing options and many machines which do all these different types of tests. The next generation sequencer, we have the Illumina platforms in the lab, we have a pyrosequencer. As I say, we have real-time PCR machines, digital PCR, and we also have FISH microscopes and workflows to analyze arrays. So how do we choose which test we will do? There are a number of aspects we have to consider when we're choosing a test. We obviously look at cost and how long the test takes to run. Some of these machines run in minutes or hours, some can take a day. But one of the key things that we would look at when we're deciding which test method to use would be the balance between how many mutations we want to detect, how many genes, what's the spectrum of mutations in your in this case, cancer of interest or your tumour of interest, and then also how low level your limit of detection needs to be. Do we need an incredibly sensitive low limit of detection when we're looking for really low level, potentially resistance mutations, or are they generally present in a higher level and therefore we don't need quite such low levels? And this kind of shows some of those platforms that we use and where they fit in this balance between limit of detection and sensitivity and how many mutations we can detect. We've got digital PCR, which has a very, very low limit of detection, but usually can only pick up 1 or 2 mutations each test. We have some real-time PCR, the COBAS assay, which again has a good low limit of detection, but will only pick up a few changes. Pyrosequencing, again, you can sequence a slightly bigger region, but your limit of detection is subsequently a bit higher. With the advent of next generation sequencing and the development of workflows, particularly around cancer panels and working on tumour material, these developments have actually led us to a position where we have an assay now where we can look at many hundreds of genes all at the same time and still have a very low limit of detection. And we can regularly go down to 4% of the mutation in the background, and down to 2% when we are targeting certain regions. So those are the kind of techniques we use. So a little bit about the input and what we're actually analysing and where this comes from, and all those factors that affect how well the test or which test we can use. So all our work in genomics starts with the extraction of DNA or RNA. As we know, the majority of tumour tissue is formalin fixed blocks. It's very good for the histology preservation, and we can store the samples for a long time in this format. Unfortunately, the formalin fixation does result or can lead to some damage to the DNA. So we have to optimize our extraction protocols and work around this. And it's useful to understand exactly what's happening to the DNA when we're optimizing our protocols. So the DNA can be damaged at any stage in the process or all stages of the processes. So when it's fixed in formalin, it can be damaged. The storage, so samples which have been stored for many, many years naturally start to degrade and accumulate damage. And then when we actually start the process of DNA extraction, we have to break the cross-links between the proteins and the DNA, and this can result in additional damage. And we get a variety of different types of damage within that DNA. The majority of the damage just results in fragmented material that we can't actually sequence and analyze. But there are some forms of damage. You get deamination of the DNA, which actually changes the bases. So we get artifactual changes, which can look like real variants or mutations. So we always have to take that into account when we're doing our analysis. There are things that we can do with the material prior to the DNA extraction that can help and optimize and help with a more successful genomic analysis. One of those is some of those factors which have started to be published about the treatment of formalin-fixed material for genomic analysis. One is to keep the formalin, so the formalin should be fresh, and they recommend a 4% neutral buffered formalin. The control of fixation time, where possible. We know extensive fixation times will degrade the DNA more. We want to do our analysis preferably on freshly cut sections. So every time there is a request, we'd ideally want to go back to the block and cut some fresh sections for DNA extraction. We need to avoid cross-contamination. We're working in very low levels of limits of detection, trying to detect often very low levels of mutation within a background of either normal cells or low amounts of tumour clones. And so we will pick up if there's cross-contamination with other tissue from other blocks or other patients, there is potential to lead to incorrect results. And so maintaining that avoiding that cross-contamination, we're looking for working in a clean area with clean microtomes and sterile clean plasticware for transport. Just a little note about decalcification. We know there are often samples which need to undergo decalcification. We just would like to mention that acid decalcification is very degrading to the DNA, and we can very rarely extract DNA which is suitable for analysis if the sample has undergone acid decalcification. There are methods for decalcification using more neutral solutions such as EDTA and using a longer time period, which are better for genomic analysis, but obviously there's a balance between the decalcification requirement in histopathology and whether the genomics is needed and how it's going to impact any downstream testing. I also just wanted to mention RNA extraction. So a lot of the work we do is in DNA, but we also extract RNA. So RNA extraction we use for the detection of structural variations that create fusions. So this is when you get a structural change where two genes in different parts of the genome, often on different chromosomes, will come together in a rearrangement, but then they will be transcribed into a fusion transcript. And by looking at the RNA, we're not just looking at the structural change in the DNA and the actual chromosomes, but we're actually looking at the ones where it's more likely to create a functional oncogenic fusion transcript. So we take the RNA, we extract that RNA from the tissue in a similar way to the DNA. We then actually convert it back to DNA, convert it back to cDNA, and we'll then do a next generation sequencing analysis in a similar way to the DNA. And we will probe for both sides. So our libraries and our NGS panels will look for both sides of those fusions so that we can see exactly which two genes have fused together, which allows us to do more informative interpretation as to what that rearrangement might be doing in the cell. So those are the kind of materials we extract. A few slides a little bit on how a pathology review before the samples come to genomics can really make a difference, and how when we're asking for particular information, how that information is used and what difference it can make. As I mentioned, all our genetic tests are based on DNA or RNA. The number of neoplastic cells is really important for us to understand for when we're doing our interpretation and analysis of our results. When we nearly always have a mixed population of cells, some will be non-neoplastic, some are neoplastic, and understanding that proportion is really useful. When we're looking at samples, it's useful to understand that necrotic cells have very few nuclei, so we will unlikely to get much DNA from them. The same goes if a sample has lots of large cells with very few nuclei, again, less DNA, but areas with small cells and many nuclei often give us an increased yield of DNA. So knowing which bits of the tissue and what those tissues and the cell looks like can be really useful. We're all very aware of heterogeneity within pathology blocks and within tumours. And this was an interesting study that came out of an EQA validation process, where they took sections across a 1,000 micrometer block, and they took 12 sections across it. It was a block which was known to have a KRAS mutation. And when they analysed blocks at different levels in this, different sections at different levels in this block, they actually saw that the mutation was only detectable in some of those sections, showing that there was quite a lot of heterogeneity through the block, and you can't always be sure that any changes are consistent in every single section when you're looking between the top and the bottom and across that. And it just highlights that when we're looking and doing that pathology review and looking at the proportion of neoplastic cells, it's really important to understand and try and do that assessment on a section that has come from close to the sections that will be sent for genomics, so we know we've got a true representation when we're doing the genomic analysis. This next bit I just want to talk about a little bit is about macrodissection. We often mention the possibility of doing macrodissection on these samples to try and enhance the amount of tumour material that we're testing. There's no doubt that macrodissection is really useful and can increase the sensitivity for our tests. So if you look at this diagram, if we scrape the whole section, and only a small proportion of it were neoplastic cells, we've potentially got a mutation level of less than 10%. And this is often dropping below the limit of detection of some of our assays. However, if we'd been able to scrape just that small piece of tissue that was enhanced for neoplastic cells, the mutation level would be over 30%. and again, well within, and then it would be well within the range of our techniques to detect. To do macro dissection, and I think it is important for people to understand that we are talking it is truly macro dissection. We're doing it manually with a scalpel. This is not micro dissection. We're unable to pick really, really small areas of the slide. It is just scraping material off a slide with a scalpel. And also, when you do the macro dissection, we are reducing the amount of tissue. So we are going to be reducing the amount of DNA that then is available for testing. So we should really only do it when it's really going to make a difference. And the difference is often we say around that 20% neoplastic cell mark. So if the overall content is well below the 20%, but you can macro dissect it to enhance those neoplastic cells so that it will be greater than 20%, then it really is worth looking at macro dissection. And to do that, we do need a corresponding H&E slide, or an image of the H&E slide would be fine, so long as we're able to line it up with the slides and the sections that come with it. So just to go a little bit more detail and show how the macro dissection helps. If you're sequencing material that is all one population of cells, we get a nice pattern, we can see every base. If our sample was all tumour cells and they had a mutation, that would only be in half of those cells, or only half of the signal would be that mutant. And you can see right in the middle there, pointed out by the arrow, you see those two peaks showing that there's two bases at that one location, indicating a mutation. If we take what is more representative of a tumour sample, so we'll have a mixed population of normal and tumour cells. In the top diagram, you've got a 30% tumour content, and you can see that peak under the arrow, but this time it's much, much smaller and quite hard to see, but definitely there. When we take that down below 20%, you can see that peak really drops off, and the actual chance of missing that change is much higher because the peak height is so small, with the number of cells, neoplastic cells, being so small. This is an example of a case we had a number of years ago, which nicely demonstrated the benefit of macro dissection. These images are pyrosequencing graphs. You can see the sequencing bases called underneath, and a peak at each base shows that's the sequence that we can see. And we're looking at a wild type sample and a sample which has a BRAF V600E mutation. And you can see some additional peaks in that lower diagram. This was our patient's sample who was run on the pyrograph. These are the peaks that we would look for to see if there is a mutation. And they do look pretty flat, but there was a slight hint in this case. So we were unsure and we weren't totally convinced there was nothing there, but there was certainly not enough of a change or enough of a peak there to call any change or call that mutation. We went back to the original sample and saw that it could have been macro dissected, and there was a region which was enhanced for tumour cells. So we macro dissected it and re-extracted it and then reran it, and this is the difference. That mutation was suddenly much more obvious and we were able to detect. So as I've mentioned, macro dissection is really, really useful if we're going to cross that limit of detection threshold for a lot of our genomic analysis samples, which is around 20% neoplastic cell content. So I've talked a little bit about the importance of neoplastic cell content assessment and how it impacts the tests we use, and understanding our limit of detection, and whether when we don't see something, whether it's truly that it's not there or it's just at too low a level to detect. You'll notice on our forms, we ask for a little bit more detail than just is it above or below 20% neoplastic cell content. These categories have recently been added to our new forms, and they've been added because they correspond, partly because they correspond to the tissue IEQA GenQA scheme, which is now running for neoplastic cell content assessment. But they also have a use in the lab. So the The 1 to 5 and the 6 to 10 don't make much difference to us in choosing a sample, although they can help with analysis. Anything between 10 and 20 is useful because we have some assays and some techniques which have that slightly lower limit of detection. Anything above 20, we would do on next generation sequencing panels where we can. So you might ask, why do we need to understand how much it is above 20? When we start doing additional analysis, and something we're working on at the moment, which is copy number analysis and copy number detection from next generation sequencing data, so we can run the next generation sequencing and not just get the small mutations out, but we can also look at copy number. But when we start to do this analysis, the neoplastic cell content assessment and a more accurate idea of how many neoplastic cells there are becomes more important. You can see from this graph that as the neoplastic cell content goes up, the number of target copies that you can detect comes down. So we often think of this is around amplifications, gene amplifications we're detecting. We often see 10 copies as a kind of threshold that potentially is then significant and causes an impact in the cell. And you can see there, to reliably pick up a change of 10 copies, an increase of 10 copies, you need around a 50% neoplastic cell content. And you certainly need above a 20% neoplastic content to pick up anything, any change above 20 copies, which is the significant amplifications. When you drop below that, it becomes very difficult to truly understand what the amplification is in the sample. So that neoplastic cell content is becoming more and more important to allow us to do more and more genomic analysis and do it accurately. Next area I'd like to just present and talk a little bit about is a recent development in the laboratory. So we're currently processing over 2,000 solid tumour samples a month. That's a huge amount of extraction that needs to go on, and it's currently all done manually. We've been working to validate and optimize some automated extraction machines, and this is the one that's in our lab currently. This is the QIAsymphony, to see if we can run a more efficient extraction and help us process all these samples in a more efficient but also a quicker way. So the initial work from this validation series, I just wanted to present some of our findings. So the graphs in blue are our old or our current manual extraction, and the sections in orange are the new QIAsymphony extractions. So the initial work we did showed that the QIAsymphony tended to give us more output of DNA, which was very, very encouraging. Of course, it's not just about how much DNA we get out, it's the quality of the DNA. So we've done a couple of downstream tests on that extracted material. The graph on the left that shows a DIN, which is a DNA integrity number, which gives you an indication of how good quality, how fragmented the material is, and how likely it's going to go down for successful sequencing. And you can see that the QIAsymphony, again, we get a higher DNA integrity number, which indicates a better quality, less fragmented material. And then when we go on to sequence that, we see that we get better sequencing coverage. So all in all, the QIAsymphony DNA extractions seem to be giving us better quality material, which is suitable for DNA sequencing. However, one of the things that we did notice is, particularly with the smaller samples, because this is an automated machine, you often do see some reductions in yield, and a lot of those smaller samples that we get, we actually saw a reduction in the amount of material. We still got that increased quality, but we were seeing this lower amount, and we felt that for those smaller samples, this could have a significant impact on whether we'd be able to go on and test them. So what we've done is some further optimization, and we've looked at different lengths of digestion of the samples and different lengths of time of removing the paraffin wax. And And what we've shown is that if we go for a slightly higher input of material, we get a much better output and a higher concentration of DNA coming out. So we've been looking at our current requests, which are 5 sections at 5 micrometers, compared to what happened if we doubled that. So we had 10 micron sections. We'd still keep the 5 sections, but with the 10 microns, whether that brings that yield up to a level when we can go on and test all of our samples. So that seems to be as I say, we're in the final stages of validation, but it looks like if we can just increase the material put in, we'll get a good quantity of DNA coming out of a better quality, which will hopefully help with downstream genomic analysis. So we're hoping to move over to this new extraction platform from the 1st of April. We would like to ask and get some feedback, and the proposal would be that when we move to our new extraction platform, this new automated machine, that we increase the amount of sections that we're being sent. So we'd ask for, instead of the 5 times 5 micron sections, we'd ask that we're sent 5 10 micron sections. In the same way as curls or sections in a tube, or slide mounted with the corresponding H&E if they need macro dissection. We do ask for 2 tubes, and this is to allow us to do DNA and RNA analysis. And as the majority of tumour types now have indications on the test directory for small variant detection as well as fusion detection, we generally ask where possible for 2 tubes for all samples. An additional, I guess, best wish would be if it would be possible for these to come in a particular tube that is compatible with our QIAsymphony. That would be great because it avoids any transfer of material at our end, but that's something we can discuss individually with labs, and we can help support get those tubes into pathology labs for the material to be sent in if that was possible at all. So you'll mostly be very familiar with our request forms. These reflect the National Test Directory, Cancer Test Directory, and the tests available for each tumour type. They contain information about the samples and the sample requirements, and they also have this area for the neoplastic cell content assessment. This is to just signpost people to the National Genomic Test Directory. There's a rare disease one and a cancer one. It's a fairly unyielding, difficult to navigate Excel, but all the information is there. This is just a snapshot of the solid tumour tab. There are also sarcoma, neurological tumour tabs. And just to say that this is updated regularly. It's generally due for an update every October and April. These do sometimes slip, and there's sometimes interim updates. So it is always worth keeping an eye on the latest updates for the Genomic Test Directory as new tests come in, or if they slightly change eligibility criteria. And finally, just before we finish, I just wanted to mention cancer whole genome sequencing, as this is a slightly different process and needs slightly different sample requirements. So there at cancer whole genome sequencing, there are a number of tumours that are eligible for this. This is actually done in two centres. So the material is sent to us for extraction. We send that material away to a large sequencing centre that's a large national sequencing centre. They do the sequencing and the analysis. Initial bioinformatic analysis is done by a national provider, by Genomics England, and then that data is returned to us for that final scientific analysis and interpretation. So how do you request whole genome sequencing? So it needs essentially 3 different forms. The form on the left is the form that accompanies the sample. So it's the same form that accompanies the fresh tissue sample, the tumour sample, and the blood sample. We then have a test request form, which actually indicates what test and what indication the sequencing is for, and then there's a record of discussion that needs to be completed with the patient as well. We can only accept fresh tissue for whole genome sequencing. It's not possible to do it on formalin fixed material. There are some guidance on the volume of tissue that's required for whole genome sequencing, but generally we can get really good DNA extraction from really quite small amounts of tissue, so we're happy to try on anything. We do need a neoplastic cell content assessment where possible. It is useful to know that because of the way the whole genome sequencing is done, and because we do it on tumour and germline blood in parallel, they actually do a neoplastic cell content assessment on the sequencing data. So when it comes back and we see the data, we will get a confirmation, or we'll get highlighted if there's any differences with the neoplastic cell content from the actual genomic sequencing data. We do ask that that the samples are not fixed if they're coming for whole genome sequencing. Ideally, they should be taken and sent to us within 72 hours. If that's the case, they can just be put in a dry, sterile pot and do not need to be snap frozen. If they are going to be stored for any length of time, or that it's going to take longer to get them to us, then they do need to be frozen as soon as possible after excision, and then stored frozen. And just a note at the bottom that we do require a germline blood sample as well for whole genome sequencing. So that's the end of my slides. Thank you very much for listening, and I'm happy to take any questions and have any discussions about what we've covered today, or anything else you'd like to know about genomic analysis for cancer testing. Thank you. I'm happy for people to put hands up or put things in the chat. I've got a couple of hands. Let me see if I can see them. Hi, is it Chitra?
Hi, Chitra, yes.
Hi,
thank you, Helen. Can you also just touch upon requesting in Hive? I know you showed paper requests, which would be probably from other centers. I do requests from Hive, and sometimes I find it difficult to find the correct terminology and the panel.
Absolutely. So yeah, we have got people from all over the Northwest region. So Hive is only applicable to our MFT trust referrals. It's an online request system in the trust. Unfortunately, it's not ideal. When it was built, which is a few years ago, it was built on an old test directory, which has changed, which means some of the indications are not there. It doesn't automatically link in with our LIMS system. So whilst it's requested on Hive, it's still essentially a paper referral for us. So all we say is, if you can find the closest thing you can to the test you request, and then just add into the comments the specifics of which test you need. We're fully aware that not everything's on there, and we are working with Hive and Beaker and other areas in MFT to improve that request system. And for non-MFT referrers, we are also working on an electronic online system that people will be able to use to replace those paper requests. So hopefully we'll move away from paper at some point. Thank you.
Thank you.
I've got hand up from the Walton Centre. I'm not sure. I don't think we can hear anyone.
Can you hear us now?
Oh, yes, we can now. Yeah.
Yeah. Okay, that's great. Thank you. It's Nitika. Yeah, and we've got the whole team here. So thank you so much. It was a very useful talk. And it was lovely to hear about, the results you've got with Kaya Symphony, and that's excellent. I just wanted to understand when you said about the smaller samples that were you needing more tissue. So what is the size of when you say small, what is the definition? Is that the number of tumor cells, or?
It's very difficult to put a figure on how small a small sample is, because we find it's different depending on the tumor types. So sometimes a sample which looks like we're getting a lot of tissue isn't giving us very good yields, whereas others we get very, very small amounts of tissue, but we get really good yields. So it's very tumour dependent. If you read a lot of areas, we often have 100 cells as an absolute minimum to get DNA out for genomic analysis, because we can't do any kind of tissue quantity or volume assessments on the samples we receive. It's difficult for us to know for sure. What we can do is do individual feedback for different tumour areas where we see there are problems. So we know a lot of our prostate samples cause us a lot of problems, but that's in a well-published fact that prostate samples do cause a lot of problems with getting sufficient material for genomics. We know that the breast cores are sometimes quite challenging, whereas our lung samples, which are very, very small, often yield really nice DNA, and we can even off really, really small samples. So it's very tumour dependent, unfortunately, and very difficult to put an actual figure on volume of tissue. But all I can say is I think 100 cells is kind of an absolute minimum, really.
Yeah, no, that's really useful. Thank you. And it's just that, with CNS tumors, the samples tend to be quite small.
Yeah.
In most cases. So I think if it's rich in tumor cells, I think what I'm trying to get at, because the requirement now stated is 2 tubes of 10 micron thick, 5 or 4 sections. But of course, that's not possible each time with each sample to be sent. So I think even if we did send one, is that still okay?
Yeah, if there's a limited amount of material, we'd just ask you can send as much as you can. The reason we ask for 2 tubes is that DNA RNA difference, because we can't separate them once they're in tubes. It's very difficult to separate the sections. So if you don't need the RNA, by all means, just send one tube. If you do need DNA and RNA, but there's limited material, we'd say just put less sections in each tube. So just put 2 or 3 or 1 or 2. If you know they're really good amount of cells and lots of tumour material there, then we should be okay with less sections in each tube.
Okay, that's brilliant. Thank you so much.
Thank you. We got next Abdul.
Thanks, Helene. Very useful talk. I just have a very simple question. I tend to, every time I get this positivity with the ROS1 immuno, I obviously request the FISH and the fusion, but there are times when the tissue is limited. So if you're automatically going to be doing the RNA fusion and you're going to detect the ROS1 anyway over there. So is there any point in me actually asking for the ROS1 FISH?
I think we'd suggest that you only ask for the main reason to ask for the ROS1 FISH would if you would need it urgently, because it can be turned around quicker, or if there isn't enough material for fusion, for DNA and fusion, then you might have a couple of slides that you could send for FISH. But routinely, if we're running the fusion panel, if we've received those 2 tubes, then that should be sufficient. Yeah, it's only we do the FISH for the more urgent ones, right, just because it's a little bit quicker.
So then automatically, then I shouldn't be actually sending you another form even. Then it's because it's going to be done anyway. So yeah, I just wait for the RNA fusion then.
Yeah, there's no need to send another form unless you specifically want FISH. And if you do want FISH, we would need those slide mounted sections as well. We can't do it off the material we've received for fusion.
Sure. Okay. Thanks so much.
Thank you. Hand up, Jim.
Yeah, thanks very much. You talked about reducing the risk of DNA damage with the buffered formalin and controlling the fixation time. And you mentioned freshly cut sections. Is there a time scale that you would consider freshly cut? Would that be effective, for example, with your new automatic analyzer? It's just in terms of the processing of the labs. What sort of time would you think is fresh, and beyond what time would need a new section cutting?
I don't know actually. Sorry, just got some feedback. Okay, is that better? I don't know if anyone's actually looked and done a proper kind of assessment of how long you can leave sections cut before you analyze them or before you extract from them. We know that sections can be cut and they can take a week to get to us in the post, or sit on the bench for a day or two in our lab before their extraction, and we don't see any significant problems. So I don't think there's any problems with sections that are cut within days or a couple of weeks. I think it's longer term. So if slides have been cut and left for a very long time, I think if we're talking months plus, then we might see some degradation, but I'm not sure if anyone's actually done the studies to show what that time frame is.
Thank you.
Thanks. Sorry, I'm struggling to see hands.
Hi.
Yeah, go for
Hi,
it. Yeah.
Mishita speaking. So just my question is somewhat related to the previous question. I was just wondering if you have any information about the impact on DNA quality if lung resection specimens are fixed for more than 36 hours.
I haven't specifically looked at it. I know we had an STP student who did a project and specifically looked at lung samples and the different type of samples that we were getting for lung, and looked at different fixation impacts, but I don't know how many resection cases she had in it. I'm happy to look into that and feed that back if I've got that information. Often we find that these things aren't done as upfront studies, but we get we'll do a load of samples and we'll get a higher failure rate. And when we look into it, we find it's a particular type, and then we start to question whether it's the fixation or things like that. I'm not aware that lung resections are giving us significant cause for concern, even if they are fixed for slightly longer. I think it's often the balance between the larger tissue needs longer. Probably has, but also because it's a large piece of tissue, the formalin has less impact. But it's that balance, I think, with the formalin fixation time. But I think there's very few studies that really nail it down of how long things should be in formalin.
Thank you so much, Helen.
Thank you. I'm just having a quick look at the chat. So question, where do we get the correct tubes for the QIAsymphony? Happy for you to contact us, email us. I can put some email addresses in the chat at the end, and we can send you some sample tubes, or we can send you the catalogue numbers and the types of tubes that they are. And that'd be really helpful if people were able to use those tubes. How quickly do we need the germline blood sent to us? So I assume it's in whole genome sequencing. So again, we recommend that germline bloods are sent to us within about 72 hours of them being taken. The DNA is pretty stable, so it doesn't need to be really quick. They don't need to be refrigerated. Please don't freeze them. Just send them. Equally, they don't need to arrive with the tumour sample. So the germline blood can be sent completely independently, and we will match the two up when they arrive and are extracted in the lab and send them off together. So you don't need to worry that they're not coming at the same time as the tumour. Let's see. A question about simultaneous extraction of RNA and DNA on the same sample. Yes, that is definitely an ambition. We trialed it with the QIAsymphony. There is a protocol, and it just didn't work very well. We will be looking at it again in the future, but we will need to look at a different, probably chemistry and a different extraction platform to do it, because the one that's currently available for the QIAsymphony wasn't giving us a good enough yield or quantity, and the protocol was really difficult and manual, and it wasn't going to be time saving for us to do it. So long term, it's very much an ambition. We just need to find the right platform to do it on. So a question about where do we find the information on whether it's a DNA or an RNA test. Generally, if it's a fusion, it's an RNA test. So the National Test Directory will tell you what type of change it is. So it will say, give you a list of the genes, and then it will say structural variation or fusion in one of the columns. And if it says that, then we'll do it on RNA. everything else we do on DNA. Okay, I think that's all the questions from the chat. Are there any more questions or comments? Okay, so we will share some, oh, sorry, Lucy.
Sorry, Helen, just something just popped into my head at the last minute. So if we know it's an RNA, sorry, if we know it's a DNA test, I'm thinking about small biopsy specimens where we sometimes have to do multiple different tests on them. If it's DNA, we only need to send one tube rather than two. Is that correct?
Yeah, that's right.
Okay.
I think we started routinely asking for two because so many indications now have both, and equally we might get asked for one and then they come back a week later and ask for the other one, and rather than us getting more material cut, we felt it was easier to get it cut up front. But I agree, where there's limited material, if you know you just need one or the other, feel free to just send one tube.
Okay, that's great, thanks a lot.
Thank you. Anna?
Hi, thanks for the talk. It's a question actually relating just for MFT, because it's a Hive question. When you were talking about giving the neoplastic cell content assessment in more detail when it's below 20%, that's not on the iGene, is it? Or is it planning to appear there, or do you want us to try and remember to state it on the free text area?
So it's not on there, no. As I say, it was developed a few years ago. When we do the new development, we'll hopefully get those different categories of neoplastic cell content on there as a drop down or a selectable thing. If possible, if it's above 20%, if you're able to give us a more accurate indication, then that'd be really useful if it could be put in the comments, but we appreciate it's quite difficult. And the other thing, if it's in the pathology report, so if it's in the original pathology report, I know some path reports now have at the bottom best block for genomics and a tumour cell content assessment. We can also try and pull it out from there, although we appreciate that's sometimes not done at the same time that the sections are taken. So we'll work to get a better system, but at the moment we're just kind of working with what we've got.
Thank you.
Thank you.